University of Washington on ontology-based annotation and integration of pathway databases.
Thank you, Robert. Let me just... Oh, it's already over there. Hi, everyone. My name is Lu Wang. I am a PhD student at the University of Washington in the Department of Biomedical Informatics and Medical Education, and I am advised by Dr. John Gennari. And today I'm going to describe a little bit of the preliminary work that we've done with the folks at the Rat Genome Database Project who work on the pathway ontology, on a way to use the pathway ontology to annotate and also begin to integrate some of the data from various pathway databases. And I'm just, is there a possible way to get, like, I guess I can't get it on both screens, but that's fine. I'll just wing it. Okay, so I guess I want to start with the supposition that pathway databases should, in fact, be interoperable. This table from PathGuide shows that we now have more than 690 pathway and pathway-related resources, and that number continues to grow. And when users take these pathway data sets and use them, they often retrieve data from multiple pathway databases. And I believe that the community cares about pathway data interoperability. There's numerous standards for pathway data exchange, like BioPax, Systems Biology Markup Language, and others. And there are a lot of meta pathway resources which allow people to retrieve these pathway models from a variety of databases. And I guess you could argue with me that maybe this means that pathway data is already interoperable, but that is a debate I am happy to have. I believe that simply being able to query for pathway data from multiple databases or having them in the same language doesn't equate to interoperability or the ability for us to understand how those pathways relate from one database to another. So that's the problem that I'm interested in tackling. So ontologies, of course, are very useful tools for aligning data sets, and we believe that in this case they can be a helpful first step for improving interoperability. So this is a reproduction of a diagram from a 2001 paper discussing ontology-based data integration, and here's just a couple of approaches that people have taken and continue to take. So there's a single ontology approach where data from many databases or the schemas can be mapped to a single ontology to allow those data sets to be joined. There's also the multiple ontology approach where each schema or each data set maybe has its own schema or ontology, and those ontologies can be mapped together to each other. And then the hybrid approach where those kind of ontological schemas can then be mapped to a single unifying ontology. And for pathway data sets, I think it's sort of a mixture of the first single ontology approach and the hybrid ontology approach, because many pathway databases do have some hierarchical structure, but many also do not. So the pathway ontology, which I'm here to talk about, is project coming out of the Rat Genome Database Project, and it is an ontology of biological pathways, the goal of which is to cover all types of biological pathways, including altered and disease pathways, and to capture the relationships between them. So I think there's two important points there. One is that this ontology does seek to represent what is like altered physiology versus kind of maybe the normal biological function, and also to describe the relationships. So currently, I think the pathway ontology has, of course, a subclass hierarchy and also the makings of a part of hierarchy. So describing those two types of relationships between pathways. And of course, the pathway ontology contains some existing annotations already. It has annotations to KEGG, the NCI's Pathway Interactions Database, as well as the Small Molecule Pathway Database. And here's just an example of what one of those annotations looks like. On the left-hand side is the part of the hierarchy above the Wnt signaling pathway in the pathway ontology. And one of the instances under the Wnt signaling pathway is KEG 04310, shown on the right-hand side. And I think something I want everyone to note on this is that on the KEG side, there is some kind of minimal organization for that pathway. So there's a little bit of KEG-specific pathway organization. So maybe that's something that is a little bit, it's more structural. And maybe lends itself to a little bit of ontology alignment techniques as well. Okay, but of course, there are these annotations to 3 databases, but annotations to other databases are necessary since some of the most popular and well-known and well-used pathway resources are not currently represented. For example, Reactome, Biopsych, WikiPathways, and the list goes on. So for this project, I am going to describe a kind of annotation pipeline for the Reactome database. And when I'm saying annotations, I'm saying for each Reactome pathway, I want to find the pathway ontology classes that best correspond to that pathway. So here are some of the questions we tried to answer. Can the pathway ontology be used as a shared ontology in this particular case? So is it useful for integrating pathway data? Can we create a predictive model that somehow can be used to speed up annotation, like traditional annotation, where curators have to perform most of the mapping? And then also whether we can employ a supervised learning method to do so. All right, so as I described, I think the traditional annotation pipeline generally consists of a lot of curatorial review. So the goal is to align Reactome and to the pathway ontology, but there are many pathways in Reactome and many classes in the pathway ontology. So can a computational model be useful here to reduce the number of total annotations that need to undergo manual curation? So what do I mean by computational model? Ideally, I guess we would train some sort of predictive model that would take Reactome pathway and predict the appropriate pathway ontology class under which it fits. So maybe there could be some training data of positive and negative annotations, and then some sort of classifier, and then an output of a bunch of annotations, which a reduced set of annotations, which can then be passed on to the curators for review. And of course, our curators at the RGD, which helped with this project, are Tom, Jennifer, and Monica, some of whom are in the audience right now. So then I thought, like, where can we turn for training data? Well, because there are existing annotations in the pathway ontology, we have somewhere to start. So I extracted those annotations out of the PW, and there aren't too many of them because the databases that were annotated to are rather small. However, there are more than 800 positive annotation examples. So we took those and then sampled some negative data for our initial set of training data. And for your reference, the pathway ontology consists of approximately 2,600, more than 2,600 classes, and Reactome, the portion of Reactome that we're dealing with consists of about 2,000 pathways. So we're getting at least as many training annotations kind of on the order of the size of Reactome. For our initial predictive model, we computed mainly engineered features over pathway names, synonyms, and definitions. And the manual features, the engineered features that we computed were kind of a IDF, inverse document frequency weighted Jacquard indices over various word tokens, cosine similarity between the sparse vector representations of word tokens, bigrams, trigrams, and character-based n-grams. And then The initial models were trained using a logistic regression classifier, which is a linear classifier, as well as a random forest classifier, which is nonlinear. And then additionally, I believe that we did not have a whole lot of training data, so we tried to use bootstrapping to supplement the labeled data. So for the reason I thought this was important is because Reactome is a large database, and for some of those pathways are going to be easier to predict annotations for than other ones. So if we could take the ones that are kind of easier to predict and that we are confident that we can get right and add those to the training data, then perhaps we can also use that as an opportunity for the model to learn a little bit more about how Reactome represents its data relative to the other training databases, KEGG, PID, and small molecule pathway database. So for bootstrapping, we have our initial training data, which comes from those three databases, the existing annotations in PW. And then we sample over that training data, we subsample it, and then for each subsample, train a classifier. And then those classifiers can then be ensembled together to produce a set of voting results over Reactome. So, and then in this case, some of those results for those classifiers are going to be we're going to feel a lot more confident about those than others. So take the top X% and add that back into the training data and sort of repeat this until our confidence in those results over Reactome are no longer very high, and we stop and we train a final model using that set of training data that we've pulled out and use that as our final predictive model. So then here's we then of course had to evaluate our predictive model, and of course our annotators thankfully were there to help us do that. So I sampled a set of Reactome pathways from the results of the predictive model. And for each of those Reactome pathways, I retrieved the top 3 annotations that were coming out of the predictive model above some threshold score, which yielded about 211 total annotations to review for our annotators. And then the reviewers were asked to look at each recommended annotation. And each recommended annotation consists of a Reactome pathway and a pathway ontology class. And to mark that annotation as either yes, it is a correct annotation, as in this Reactome pathway belongs under this PW class, no, it does not, or related, which means it might not be, the annotation may not directly be to that PW class, but it may be to a related class in the pathway ontology hierarchy. And then, of course, to comment on an appropriate annotation if nothing appropriate was recommended by the system. So here are the results, at least for those 211 annotations. The raters mostly were on diagonal in the sense that they agreed. There are some off-diagonal results, but thankfully nothing was completely off-diagonal, so no one said yes, and for no annotation did someone say yes and the other person say no. The agreement over those annotations was 0.73, with Cohen's kappa over the 3 classes is 0.56. It's okay. It's not super great. But when we aggregate those over the pathways themselves, so for the 93 Reactome pathways, for 77 of those, the predictive model was able to retrieve a pathway ontology class that at least one of the raters said was somewhat related. But for 16 of them, the model performed badly, like it was not able to retrieve anything. So why was this the case? And I admit here that actually in my subsampling of Reactome, pulling out these 93 pathways, I think I probably should have randomly sampled Reactome pathways, but instead I just took a slice of it, and that probably contributed to this problem. Most of the pathways that had no appropriate annotations may not have corresponded to any classes that existed in the PW ontology already. So many were specialized immune response pathways. And if you look at the immune response pathway hierarchy from the pathway ontology, which is on the right-hand side, don't expect you to really read this. It has 35 classes in total that are subclasses under the immune response pathway. And at the finest level of granularity, some of those pathways are still of lower granularity than the reactome pathways for which we were unable to find a suitable class match. But ideally in this situation, if there is no exact class match, we would want maybe the highest granularity proximal match, but the model is unable to do that in this case. However, this does tell us that there may be a need to continue development of the pathway ontology. There may be classes that need to be added or relationships that need to be added to address some of these annotation difficulties. So I guess in summary, here's what we learned. I think when existing annotations exist, it is possible to use those annotations as training data to generate annotations to previously unseen data sets. We found that not all pathways in Reactome, in this case, aligned with classes in the pathway ontology, and that perhaps some ontology development is needed in conjunction with annotation to make that possible. And then I think the error analysis that we did is going to drive further iteration on the predictive model. There are many things that we can do here and are thinking about trying. Maybe using OPA-TVEC, it would be a good way to do that, wherever. And then there are and will continue to be updates to the pathway ontologies as more classes and relationships are added to it. And then, of course, with the underlying goal of performing data integration over these pathway databases using the pathway ontology annotations as kind of an anchor. In summary, we believe that the pathway ontology is a useful ontology for organizing pathway data. We've already started doing it, and there will be further annotations added. A predictive model can often be used to reduce the amount of manual review or for annotation that needs to happen. And of course, there's plenty more work that needs to be done on both pathway data integration as well as the development of the pathway ontology. I'd like to thank my advisor, John Gennari, thank the RGD group at the Medical College of Wisconsin, as well as the late Dr. Victoria Petri, who's one of the early developers of the pathway ontology. And then I'd love to take questions.
Okay, so we have time for a few questions. Okay, then I have a question. So I am very often a fan of external validation. So have you considered taking, for example, a metagenomics data set and determine, so basically do a prediction of which pathways are present in your microbes or in your sample, and then see which of your different databases gives different results? It might be that formally they are not properly aligned, but in real samples this never actually matters.
I see. So this is part of probably the long-term future work for me, which is evaluating whether integrating pathway data sets is going to be important to the outcomes of pathway-related analyses. I am doing some work using kind of synthetic data sets, gene expression data sets, and JACEA, where the gene sets are generated from pathway data. But this is also something I want to reproduce with, ideally, real data. But that is something to come, and which I'd love to receive more suggestions of how to do that maybe in the best way.
Last question.
Yeah. In the limited annotated data that you had, did you try to do any analysis on okay, how hard is this for even for humans to do, to make a comparison on how well the model did in comparison to, say, agreement between two humans? And the second question I had was, how many more annotations do you think one would need to, say, have a better model and probably have some sort of a standardized set to make some good progress on this?
So I guess there were two questions in there. I'll try to. I think I'll answer the second one first because it's easier. So how much data do we need to annotate for this to become useful? I think currently most of the classes in the pathway ontology there are no pathways annotated to them, just based on the number of existing annotations. So it would be, I think, at least achieving some annotations for all of the classes would be helpful. And part of the reason that gap exists, I think, is because a lot of the pathways in the pathway ontology deal with altered or altered pathways or kind of variants for which I guess like current pathway databases may or may not even label their pathways as variant pathways or altered pathways. And then the first question, I guess, was
how hard is it for humans to do the same task?
For humans to do it? I think the annotators perhaps would have a better answer for that. I don't know too much about the initial annotation procedures that were done prior to Reactome, so I unfortunately cannot give a very good answer for that right now.
Okay.
Thank you.
So let's thank our speaker again.